I am Dr.B.Kalaivani, Associate Professor, Department of Zoology, Sri Sarda College for Women, Salem. I am going to discuss about the topic enzymes. Enzymes are biological catalysts that speed up the rate of the biochemical reaction. Most enzymes are three-dimensional globular proteins, that is tertiary and quaternary structure. Some special RNA species also act as enzymes and are called ribozymes. Example, hammerhead ribozyme. So this is the structure of the hammerhead enzyme. Structure of enzymes. The active site of an enzyme is the reagent that binds substrates, cofactors, and prosthetic groups and contains a residue that helps to hold the substrate. Active sites generally occupy less than 5% of the total surface area of enzyme. Active site has a specific shape due to tertiary structure of protein. A change in the shape of protein affects the shape of active site and function of the enzyme. So the active site can be further divided into two types, binding site and catalytic site. Binding site chooses the substrate and binds it to active site. Catalytic site performs the catalytic action of an enzyme. Cofactor. Cofactor is the non-protein molecule which carries out chemical reactions that cannot be performed by standard 20 amino acids. Cofactors are of two types, organic and inorganic. So without the cofactor attached, the protein is not active. The cofactor binding activates the protein. So this is the inactive protein and this is the active protein. With the help of cofactor only, inactive protein is converted into active protein. So inorganic cofactors and organic cofactors. So these are inorganic cofactors. These are the inorganic molecules required for the proper activity of the enzymes. For example, enzyme carbonic anhydrase requires zinc for its activity. Hexokinase has a cofactor magnesium. Organic cofactors. These are the organic molecules required for the proper activity of enzymes. For example, glycogen phosphorylase requires the small organic molecule pyridoxal phosphate. So the types of organic cofactors, prosthetic group and coenzyme. Prosthetic group is a tightly bound organic cofactor, example flavins, heme and groups and biotin. And coenzyme is loosely bound organic cofactor, example NAD+. So this is the prosthetic group and this is the coenzyme. So an enzyme with its cofactor is removed is designated as haploenzyme. The complete complex of a protein with all necessary small organic molecules, metal ions and other components is termed as holoenzyme of holoprotein. Substrate. The reactant in biochemical reaction is termed as substrate. When a substrate binds to an enzyme, it forms an enzyme substrate complex. So this is the substrate and this is the enzyme. Substrate and enzyme combines to form enzyme substrate complex. Sites of enzyme synthesis. Enzymes are synthesized by ribosomes which are attached to the rough endoplasmic reticulum. Information for the synthesis of enzyme is carried by DNA. Amino acids are bonded together to form specific enzyme according to the DNA codes. So this is the endoplasmic reticulum and these are the ribosomes. Intracellular and extracellular enzymes. Intracellular enzymes are synthesized and retained in the cell for the use of cell itself. They are found in the cytoplasm, nucleus, mitochondria, and chloroplast. For example, oxidoreductase catalyzes biological oxidation. Extracellular enzymes are synthesized in the cell but secreted from the cell to work externally. For example, digestive enzyme produced by the pancreas are not used by the cells in the pancreas but are transported to the duodenum. So these are all the characteristics of an enzyme. Enzymes speed up the reaction by lowering the activation energy of the reaction. Their presence does not affect the nature and properties of the end product. They are highly specific in their action, that is, each enzyme can catalyze one kind of substrate. Small amount of enzymes can accelerate chemical reactions. Enzymes are sensitive to change in pH, temperature, and substrate concentration. Turnover is defined as the number of substrate molecules transformed per minute by one enzyme molecule. Turnover for catalysis 6 into 106 per minute. Nomenclature of enzyme. An enzyme is named according to the name of the substrate it catalyzes. Some enzymes were named before a systematic way of naming enzyme was performed. Example, pepsin, trypsin, and renin. By adding suffix ase at the end of the name of the substrate, enzymes are named. Enzyme for catalyzing the hydrolysis is termed as hydrolase. For example, maltose and water is converted into glucose plus glucose in the presence of the enzyme maltase. These are the examples for the naming of the enzymes. So lactose is the substrate, enzyme is lactase is the enzyme, the products are glucose and galactose. Maltose is the substrate, maltase is the enzyme and the product is glucose. Cellulose is the substrate, cellulase is the enzyme and glucose is the end product. So like that, lipid, starch and protein, etc. Classification. So classification of enzymes. A systematic classification of enzymes has been developed by International Enzyme Commission. This classification is based on the type of reactions catalyzed by enzymes. There are 6 major classes: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Oxidoreductases do the function of reduction and oxidation, that is redox reaction, for example, lactate dehydrogenase. Transferases will move the chemical group, for example, hexokinase. Hydrolases will do hydrolysis, for example, lysozyme. Lyases will do non-hydrolytic bond cleavage, for example, fumarase. Isomerases will do intramolecular group transfer, that is isomerization, for example, triose phosphate isomerase. Ligases will do synthesis of new covalent bond between substrates using ATP hydrolysis, for example, RNA polymerase. So, mechanism of enzyme action. The catalytic efficiency of enzymes is explained by two perspectives: thermodynamic changes and process at the active site. Lock and key model. So this model was proposed by Emil Fischer in 1894. Lock and key hypothesis assumes the active site of an enzymes are rigid in its shape. There is no change in the active site before and after a chemical reaction. Induced fit model. More recent studies have revealed that the process is much more likely to involve an induced fit model proposed by Daniel Koshland in 1958. According to this, exposure of an enzyme to substrate causes a change in enzyme which causes the active site to change its shape and allow enzyme to substrate bind. So this picture shows the induced fit model, how the enzyme change its structure to fit the substrate to accommodate. So this is the induced fit model. This is the substrate. This is the active site. So this substrate comes and bind in this active site to form enzyme substrate complex. Enzyme kinetics. It is a branch of biochemistry in which we study the rate of enzyme catalyzed reactions. So this picture shows the enzymes lower the activation energy of a reaction. Kinetics of enzyme catalysis. Enzyme catalysis, it is an increase in the rate of reaction with the help of enzymes, that is catalyst. So these are the factors affecting rate of enzyme catalyzed reactions: temperature, pH, that is hydrogen ion concentration, and substrate concentration. So effect of temperature. Raising the temperature increases the rate of enzyme catalyzed reaction by increasing kinetic energy of the reacting molecules. Enzymes work maximum over a particular temperature known as optimum temperature. Enzymes for humans generally exhibit stability temperature up to 35 to 45 degrees Celsius. The temperature coefficient is a factor Q10 by which the rate of biological process increases for a 10 degrees Celsius increase in temperature. For most biological process, Q10 is equal to 2. So this picture shows the effect of temperature on enzyme activity. Increase in temperature leads to increase in activity up to particular temperature. This particular temperature is known as optimum temperature. So after that, the protein denatures. That is during 40 degrees Celsius, the protein denatures. The enzyme activity slows down. Effect of pH. So this here also same process. Rate of almost all enzymes catalyzed reactions depends on pH. Most enzymes exhibit optimal activity at pH value between 5 and 10. High or low pH value than optimum value will cause ionization of enzyme, which result in denaturation of enzyme. So here also increase in pH will leads to increase in reaction, but up to particular pH. This pH is called optimum pH. Optimum pH is 8. After that, ionization will cause. That leads to denaturation of the enzyme. So this picture also shows the pH affects the formation of hydrogen bonds and sulfur bridges in proteins. Michaelis-Menten equation. So it is an equation which describes how reaction velocity varies with substrate concentration. V0 is equal to Vmax into substrate divided by Km plus substrate, where V0 is the initial reaction velocity, Vmax is the maximum velocity, Km is the Michaelis-Menten constant, that is K1 plus K2 by K1. S is the substrate concentration. So this picture also shows the substrate concentration. So Michaelis-Menten plot is V is equal to Vmax plus substrate divided by Km plus substrate. This is the initial reaction. This is the substrate concentration. Pharmaceutical importance. Enzymes are virtually involved in all physiological process which makes them the targets of choice of drugs that cure or ameliorate the human disease. Applied enzyme kinetics represents the principal tool by which scientists identify and characterize therapeutic agents that selectively inhibit the rates of specific enzymes catalyzed process. Enzyme kinetics thus plays a crucial role in drug discovery as well as elaborating the mode of action of drugs. Thank you.